Okay. Good afternoon, everybody. Welcome to another edition of our Matter to Life lecture series, where one week, Wednesday in a month, we have one of our fellows presenting their latest cutting edge research within the scope of Matter to Life. And this week we have the pleasure of having our new fellow, Serafina Wegner, who joined the Matter to Life faculty last year. Serafina did her PhD in chemistry from the University of Chicago, and then moved to Germany and as a postdoctoral fellow in the Max Planck Institute for Intelligent Systems and the Department of Biophysical Chemistry at the University of Heidelberg, where after her postdoc, she became a group leader and then moved to the Max Planck Institute of Polymer Research in Mainz to become an independent group leader. And since 2019, she has a full professorship in the Institute of Physiological Chemistry and Pathobiochemistry at the University of Münster. And Serafina's research focus is obviously very, very interesting to us here at Matter to Life. She works on bottom-up synthetic biology, specifically tissue engineering and light-controlled minimal cellular system and photoswitchable systems. And today we're going to listen to some parts of her research, hopefully. And before we move on to the research and her exciting results, we have a few icebreaker questions. And the first one is, first, welcome, Serafina.
Thank you.
Why did you decide to apply and become a part of the Matter to Life?
Thank you for this nice introduction. And the question, for me, I think it was a very obvious step to apply to the Matter to Life program because I started my group in an interdisciplinary network that was on bottom-up synthetic biology called MaximBio. And many of the fellows within the Matter to Life were already partners back then with me, and I just wanted to continue this interdisciplinary research that I had done all along in my group. So for me, it was kind of coming back home.
Jen, a quick follow-up is how does your research engage with the interdisciplinary perspectives of Matter to Life?
I think you will see some facets of it also in my talk, but we like to have minimal systems to understand how cellular functions arise. But then we can use the same concepts and implement them into cellular systems and question really our understanding and see if what we observe in the minimal system and the cellular system match. And I think this switch between more complex and simple system really gives us additional insights that we would not be able to get if we were just studying one. And I think this really fits into this concept of matter to life. Yeah.
Totally. And the last one, if you weren't a scientist, what do you think you would be doing?
Oh, I think this is a very challenging question. I think I never want to be bored with what I'm doing. And being a scientist is, of course, a pleasure of doing what I love to do every day and getting paid for it. I like this creativity aspect of it and creating things. And so I think I could have been an engineer or an architect or maybe even an artist, kind of a painter or a sculptor. But yeah, those would be my options, I think.
Well, I'm happy you decided to become a scientist. Yes, and for those who are attending, and we have Serafina will give her talk, and at the end of it, there will be a Q&A session. So if you have any questions, there is a Q&A bubble at the bottom of your screen at the Zoom, and you can always type it in that, and we would read it out and answer that at the end of the session. And with that, I hand over the stage to Serafina, and I look forward to hearing her talk and her creativity in science.
Yes, thank you very much. Yes, let's pick up this creativity aspect and really think about what we can create. And I really like this kind of concept of synthesis that chemists bring to the table, that you can, of course, observe things, and that brings you understanding. And that's kind of how biology has approached the concepts of cells. But chemists will tell you that equally important is synthesis. Synthesizing systems allows you to understand them, build them, engineer them. And that's why I kind of like to take this idea of synthesis and bring it to my new synthetic target, the cell. The cell, of course, is just a micrometer-sized object. Yeah, if you're talking about a bacteria, it's like a micrometer, but a mammalian cell can be 50, maybe 100 micrometers in scale. But if you think about the molecular complexity that is housed within these few micrometers of space, it's enormous. So we have 3.2 billion base pairs of information. This codes for about 20,000 proteins. These are then also modified after their synthesis, and then you have a bunch of other biomolecules. And somehow all these molecules come together and give rise to this phenomenon that we call life. And we so far know of life only on this planet. And us being here is evidence that there has been a transition, that non-living material has becoming living. And that's the motto of this Matter to Life school. And that's also really one of the core motivations of synthesizing a cell for us. And if you look at how synthetic chemistry has looked at synthesis of complex molecule, you can say that it's not just about the final target, but on the way of synthesizing the cells, we will have intermediates. And these intermediates will be more cell-like and also will enable new technology. At the same time, we will also develop a lot of new methodology because this is a whole new type of synthesis that we are doing that is different from organic synthesis or inorganic synthesis as we know it. And this involves non-traditional synthesis methods such as microfluidics or cell-free protein expression. So there's also a lot of methodology that needs to be developed on the way. And finally, what I also really am excited about in this research space is that we can go beyond what evolution has discovered, and we can introduce new features in our synthetic systems and maybe also transplant them into living systems so that we can create new types of biology. And so all of this is for me a core motivation for running this research program. So yeah, the idea is really to do a retrosynthesis of the cell, break it down into small parts. They can be of biological origin, or they can be synthetic parts that normally natural cells don't have access to. But what is important is that we can assemble them in a precise manner so that we really understand how each molecule is interacting with each other. And then we can produce systems that more and more mimic living cells. And in the end, the dream is that we get to this transition from non-living material to life. When we were starting this program, something that in my group we quickly realized is that a lot of functions in the cell arise from spatiotemporal regulation. Okay, this is a big word first, but let me break it down. A cell is chemically non-homogeneous. There is different chemistry going on in different spaces and at different times within the cell. And this inhomogeneity of chemistry is essential for living cells to operate at the subcellular scale, but also at the multicellular scale. And this is the core motivation for us to regulate systems so that we can mimic this non-homogeneity in space and time. And this can be for very different processes, like protein localization. You might need the protein to be precisely localized at a point at a certain time for its function. Or if your cell is moving, it needs to have different chemistry going on in the front and in the back. The same thing, of course, also holds true when you think about gene expression. You want to activate certain genes at certain times, and this is the basis of differentiation. During cell division, you have certainly a highly coordinated action. This is also true for cells cells communicating with each other and also how tissues organize. So how can we introduce spatial temporal regulation into cellular system? Well, the answer for me is light, because light gives you supreme spatial and temporal control. I mean, I think one has just to first realize how great the spatial temporal resolution is. Temporal resolution means that you can instantly deliver your input at the speed of light, literally. There's nothing faster that you can deliver than light. And at the same time, you can also remove it at the speed of light, which I think is equally important. You just turn off your light. The second important point about the spatial resolution is that you can have a diffraction limited spot in resolution. So we are talking about sub-micrometer, down to sub-micrometer resolution, and that gives you subcellular resolution, and that's the kind of scales that we are aiming for. And at the same time, it's very tunable, so you basically can titrate your system like you would with a chemical, but by just changing the intensity of light. And this makes already light a very unique way of manipulating, of controlling systems. But there is more. Light is not just one color, but we have a whole rainbow of colors that we can use. And we can use different colors of light to address different functionalities. The way we do this is by using photoswitchable proteins. So these are proteins that respond to visible colors of light, and they can have different types of actions. Most of them here represented in the scheme are light-dependent homo or heterodimerizers. And these mostly respond to blue or red light. And the other nice thing is that these interactions are reversible. So when you turn off light, they will decay and you can go back, and that allows you to bring in dynamics. At the same time, these are very specific protein-protein interactions that can also work in complex environments such as the cell, because they actually come from biology. So they are made to operate in the complex environment of the cells. And a very important final point is that the intensities of light that you need are very low. We are talking here about low intensity visible light that does not damage the cell, does not cause phototoxicity. In fact, some of these proteins have evolved for organisms to avoid light, just to tell you how sensitive these systems are. And that's why we want to implement photoswitchable proteins, both in the context of synthetic cells as purified proteins, but at the same time, we can use them in cellular systems as genetically coded building blocks and implement them there. And so today I thought that I would focus on one research area in my group where we are looking at controlling cell-to-cell communication between synthetic cells. And I think this is a very nice feature of cells, as it's also kind of relates to what we are doing, because also we are communicating. So currently I'm the sender, acting as a sender, and you online are the receivers. And how does this communication work? Well, I'm sending out a signal. This is now sound waves. And hopefully you are listening to me and receiving the signal, and this leads to a reaction by you. And of course, how well this communication works very much depends on many factors. And it's very important when and where we are communicating. One important regulator of communication is signal amount versus distance. So if we are communicating over large distances without having an amplifier, so now the internet, we need a lot of signal. But if the sender and receiver are in close proximity, then you can operate with much less signal. And this is a concept that is also true for cells communicating with each other. The only difference is that they usually use chemicals for communicating with each other and not sound waves. So how can we mimic this in a synthetic system? Well, we can use lipid vesicles. So these are giant unilamellar vesicles, a lipid bilayer basically. And this is a vesicle that is the size of a mammalian cell. So we are talking about 20 micrometers approximately. And our sender cell here is filled with a chemical signal, which is calcium. And we also have a receiver vesicle, which has a receptor for calcium. And in this case, this is a calcium-sensitive fluorescent dye. So now if the sender releases its signal through the addition of an ionophore, which basically makes the calcium leak out of the membrane, this signal can diffuse to the receiver, but if the receiver is too far away, the signal will be diluted before it is perceived. On the other hand, if the sender and receiver are in close proximity, then the receiver can get the signal and can respond to it through a color change. And we asked ourselves if we can control basically the proximity of the sender and the receiver using light. And for this purpose, we used a protein called islet, which is blue light activatable. And upon blue light activation, it binds to a partner called nano. And thereby, we can basically induce adhesions between the sender and the receiver. So this was our initial design. And it is how it looks under the microscope. So the center vesicle here is in green with a green membrane dye, and the receiver with a red membrane dye. And when you add the ionophore, you can see that the receiver lights up in the green channel from its side. So it has received the calcium signal. And now you can ask the question, is this light dependent? And the answer here is yes. The receivers respond more to the calcium signal when we do the experiment under blue light compared to the dark. So basically with light, we have controlled who is in proximity and in receiving range. The other part is that you can maybe not just have one receiver, but you may have two potential receivers. And how can you regulate who is getting the signal? And so for this, we have thought, okay, can we basically rearrange the organization and control which receiver is within signaling range? And so for this now we use two different photoswitchable proteins on the surface of our sender synthetic cell. It's islet and Phi-B. And in this case, I should mention that this is not a lipid membrane, but it's a protein-based membrane, so that it is leaky for molecules up to a certain size. When we mix the one sender and two receiver populations with each other in the dark, they randomly distribute. However, if we start to illuminate with red light, the sender binds to the receiver one population, which is functionalized with a complementary binding partner, and thereby the sender and the receiver one get into close proximity. And the opposite is true. If you illuminate the system under blue light, then the islet nano interaction is activated, and thereby the sender is in proximity of the receiver too. This is just the binding, the adhesion, but how about the signaling? And so here we built a different signaling cascade, which is based on DNA strands. So here the sender is activated with an input strand, and there's a strand displacement reaction, which leads to the release of a DNA strand, which is here this Q1 strand. And when the sender releases the Q1 strand, it lights up in the green channel. So you can know that the sender has been activated. And then if the receiver here is in close proximity, the Q1 strand can go into it, and there's a second strand displacement reaction that takes place, and then the receiver one population lights up in the red channel. So what happens if we do these experiments under dark versus red light? You can see that in the dark, the sender is activated, so it lights up in the green channel, but the receiver one is at some distance and it does not get the signal. The opposite is true when we do the experiment under red light. We see that the sender is first activated in the green channel, and then also the receiver is activated in the red channel, so showing that it has been activated. And so you can do this with the receiver 2 population the same, just that now the communication is blue light dependent. What is of course more interesting is if what happens if you mix all three populations. So you can see that in the dark, only the senders are lighting up in the green channel, but the receivers are not increasing in the red channel. When we do the same experiment under red light, you can see that the receiver 1 population lights up in the red channel, but the receiver 2 population, which is labeled with a blue fluorescent dye, is basically not changing its color. And if you do the experiment then under blue light, you can see that some of the receivers are now turning purple, showing that the receiver 2 population got its signal. And so you can do this, of course, on a quantitative level, and you can really see that depending on which light illumination the system is exposed to, in the dark, no receiver is activated and there's non-responders. Under red light, the receiver 1 population is activated, and under blue light, the receiver 2 population is selectively activated. So this is the communication between cells. But cells, of course, also have to perceive signals from their environment and then respond accordingly. And this is not just one signal, but cells are exposed to all types of signals in their environment, and they need to react accordingly. And we thought, can we generate a system where we have different chemical inputs that lead to different chemical outputs? And for this, we, for once, did not use light, but we used metal ions. that we wanted to transport into our synthetic cells differentially. And for this, one can use small molecules called ionophores that can very selectively transport different metal ions across membranes. So if we first identified one certain ionophore, which I will call ionophore A, and this one selectively only transports nickel ions, but another ionophore, two other ionophores that I will call ionophore B and C, do not transport the nickel ion across the membrane. As you can see, the transport of nickel ion leads to the quenching of the internal fluorescence of the vesicle, but in fluorescence in B and C, there is not such a reaction. Then we can, of course, look for other metal ions. So ionophore B is very selective for copper ions, and the ionophore C is very specific for calcium ions. So depending on which ionophore is added, we can transport differentially a different metal ion. This is nice, but this is just like a metal ion, and you have not really activated a chemical reaction inside your system. To expand on this, we thought that we can use metal ions to activate dormant metalloenzymes. Metalloenzymes are actually very important. Like more than 1/3 of all enzymes are believed to be metalloenzymes, and they really need their specific metal cofactor. They will not work with a different metal. So for the nickel ion, we thought that we could use the enzyme called urease, which is a nickel-dependent enzyme. And so we encapsulated it in an apo form, and upon transporting nickel ions, we could activate it and thereby activate this reaction, the decomposition of urea into ammonia and bicarbonate. And this thereby increases the internal pH, which we could visualize with a pH-sensitive fluorophore. For copper, we chose an oxidase. It's a galactose oxidase, and through the activation of galactose oxidase with copper, we could generate hydrogen peroxide. So we activated a different chemical cascade. And finally, for calcium, we encapsulated apophospholipase, and only upon the transport of calcium into the vesicle, we could induce the lysis because the phospholipase basically chews up the phospholipids that the membrane is made out of. So, okay, it's nice. We could activate one of 3 enzymatic reactions, but we thought, okay, can we sequentially activate different enzymatic reactions that within a synthetic cell? And here we were caught by a surprise. In fact, when we first add ionophore A, it transports nickel. But if we first add ionophore B or C and then add ionophore A, nickel is no longer transported. And it's also the same if we first add B and then C and A as the final ionophore, so third, there's also no transport. So it seems that the addition of another ionophore is impairing the action of the nickel ionophore. And this was not just true for the nickel ionophore, it was actually true for all 3 ionophores, that they had a negative impact on each other's action. And only the ionophore that was added first was able to transport its metal ion. There was a little bit residual activity with the second one, but after that was really not active anymore. So this reminded us of a negative feedback, so that we had like 3 possible dormant enzymes that we could activate, but it really mattered in which sequence they were exposed to these signals. And to understand this, we did collaborate with a professor at my university, Andreas Hoyer, who did MD simulations of these ionophores within the membranes. And what he found is that if you have one ionophore A, for instance, it can move within the lipid bilayer and it can flip between leaflets, and that basically is what allows it to transport the metal ion. However, if we introduce another ionophore, such as ionophore B, the movement of this ionophore A was much more limited. And the same also was true for the presence of the ionophore C. So you can see that the ionophore A is much less frequently transitioning between the leaflets. And this is because the ionophores basically are stacking to each other. So there's pi-pi interactions that are stronger than the hydrophobic interactions with the lipid tails. And this is what leads to this negative feedback that we were observing. And so what we then could do, we could use this to change the activation profiles with ionophores. So when we first add ionophore A, it activates the urease, all fine. Then we add ionophore B, and the urease is hardly activated, only on a very low level. And then we add the ionophore-C third. And I remind you, this is the ionophore that led to GOV lysis before. And the vesicle is completely fine. There is no degradation of the vesicle. So it has become insensitive to the presence of ionophore-C. We can, of course, then switch things around. So when we add ionophore-A first and then ionophore-C second, we can see that there is actually some residual phospholipase activation, and this leads to some leakiness of the membrane. So we have now a leaky membrane because we added the ionophore-C second. And ionophore-B being added third, it has no more effect. And then you can flip the order. So you can first also activate the oxidase by adding the ionophore-B first. And then you can add ionophore-A, and you can see that urease is hardly activated. And again, ionophore-C is completely irrelevant at this point. And just to make it complete, you can also add ionophore. After ionophore-B, you can add ionophore-C, and you can see that your vesicles are getting a little bit leaky. And then ionophore-A has no action at all anymore. And so what this reminded us is the adaptive activation of a pluripotent synthetic cell. So initially, we have a pluripotent synthetic cell, which has 3 potential dormant activities. And the addition of the first ionophore basically seals its fate. That's the enzyme that determines which enzyme is being activated the most. And then the second ionophore basically then only has a minor action. So it can lead to a subspecialization. So it can lead to from a pluripotent to a differentiated state. And the third ionophore is basically irrelevant at this point because our synthetic cell has fully differentiated, and the third ionophore has no action at all anymore. So it has been desensitized to the third ionophore, if you will. So far to communication with the outside. But now let's think about generating new biology. And so far, all our interactions have been based on chemical communication. But we asked ourselves, can we actually use light instead of chemicals as a signal that is transferred from the sender to the receiver? Why should we think about this? Well, actually in biology, there is light-based communication, but at the macro scale, and it leads to a lot of very interesting behavior, from fireflies finding their mates to anglerfish luring their prey, or for some squids that can confuse their predators based on light signals. But at the cellular scale, light-based communication has not been described. And that is despite its potential benefits. Because if one thinks about light-based communication, it's completely different from the existing chemical communication modes, and that would open basically a new dimension in communication that we can freely program and would not perturb the chemical communication at the same time. It also is very fast because the signal travels at the speed of light, and it can cross membranes, so you don't have to have transporters anymore. And you can even think about communication between physically separated cells, so you don't even have to have chemical connectivity. It's not affected by things like diffusions or flows or turbulences. And if we understand how to do this well, we could even code for different signals using different colors of light. So we have the potential of having multiple programmable communication channels. So how should one go about this? Well, first you need to think about how can you generate a light signal from the sender. And then you have to think how you can translate this light signal into an action through an extracellular light responsive element. Generating light from a synthetic cell is not complicated. You can encapsulate an enzyme that catalyzes a bioluminescence reaction, and you can generate photons from inside a vesicle. That's is rather straightforward. Then if you think about the light responsive elements, there's kind of 2 considerations. One is that you have to think about the spectral match of the 2. You can see that the luminescence that we are generating is in the blue, and that matches perfectly with one of our blue light responsive proteins, islet. And then the second question is, do you generate enough photons for activating this photoreceptor? And so for this, we immobilized islet on the surface of a vesicle and added a complementary interaction partner that is fluorescently labeled. And we could see that as we are generating photons, there is recruitment showing that the islet is activated. And at some point, the substrate is consumed and we are not generating photons anymore, so the interaction is reversible again. So that's fine. But how to then translate into interstellar communication? Well, we can also use the bioluminescence to generate proximity between the sender and the receiver. And in fact, in here you can see that we generate direct adhesions between the red and the green labeled vesicles, so between the sender and the receiver. So now let's translate this into some behavior. So we were inspired by the anglerfish that lures its prey through its light and then upon proximity goes for a kill. And we asked ourselves, can we generate basically a microscale analog of this? And so for this, yeah, you can basically now continue to add chemical components to realize this type of action. So I already showed you that we can trigger adhesions upon luminescence generation. And now we are adding pores in the form of alpha hemolysin pores, but these are initially blocked. That's very important. And only at the site of adhesion, the blocker is removed so that a secondary chemical signal can be transmitted from the sender to the receiver upon adhesion. And this now is calcium, and the calcium activates the phospholipase that is inside the prey vesicle. So basically, we have a glowing predatory vesicle that is there to lyse the prey. Does this work? So first we left out the phospholipase and just showed that we have contact-dependent calcium transfer. So you can see here the red vesicle is a receiver vesicle, which has bound to a luminescent sender, and it says light up inside in green, showing that the calcium is received. And you can see here distal red vesicle that is not binding to a sender does not have this type of action. So we have contact-dependent calcium transfer. And then we can look at this under the microscope in real time, and you can see that the predator has captured the prey, it captures the calcium, and it can then lyse selectively the prey vesicle. And as this is a molecularly programmed system, we can really question if each component is performing as it has been molecularly programmed to do. So we mix equal numbers of sender and receiver vesicles, and you can see that selectively only the prey population is going down when all components are present. And if you leave out any one of them, the prey and the predator will coexist happily. And this light-based communication is interrupted. So yes, light-based communication does work at the microscale. But is more, one can also needs to think about self-regulation. So there's some communication, but at some point you run out of signal, and then there has to be a way of leaving. So basically when the communication is over. And for this type of self-regulation, we also thought about, can we couple proximity to the production of the chemical message? And so here in our design, the chemical message is hydrogen peroxide that is sent from the sender to the receiver. But we wanted to find a way that once the chemical signal is over, the receiver can detach. And for this, we use a different type of luminescence reaction that uses now chemiluminescence. And chemiluminescence is a famous reaction, is the reaction between hydrogen peroxide and luminol catalyzed by HRP. It generates light, and thereby this chemiluminescence was engineered to activate the adhesions between sender and receiver. And once the hydrogen peroxide production is down, basically there's no more light production, and this then should lead to the reversible dissociation of islet and nano. So how does this work? Well, we can look for, first of all, if the chemiluminescence is sufficient to generate adhesions between sender and receiver. And yes, indeed, this is the case. And then you can see if with chemiluminescence, you can also transfer the hydrogen peroxide signal to the receiver, which is here in red. And if a receiver has received the chemical signal, it lights up here in cyan. And again, you can see that if you leave out the adhesions or the chemiluminescence, that there is no communication. Then we can also see that for how long we are producing light, and we see that we are generating light here for about 2 hours, and at the same time, it is during this first 2 hours, the interactions between sender and receiver peak, and then over time they dissociate again, really showing that the proximity is coupled to the signal generation, and once the conversation is over, sender and receiver can go separate ways. I also would like to then extend this concept. So until now, everything was in synthetic cells, but I also said that we like to bring in the same concept into living cells. So can we use light-based communication to have communication between living and synthetic cells? And for this, I'd like to show one example where we would like to target neutrophils with synthetic systems. So neutrophils are part of our immune system and they generate hydrogen peroxide when they are activated. And we thought, okay, can we use this hydrogen peroxide as a fuel to generate chemiluminescence? And then this chemiluminescence could be used to activate a photoswitchable protein. And in this case, the protein is called BCL4. It has a little bit of a different mode of action. Once it's activated, it binds to negatively charged surfaces, membranes, and this is also true for the neutrophils. So we basically want to target BCL4 to activated neutrophils using this light-based communication. So first, I can show you, we did a synthetic model. So we now have here again a vesicle with the catalyst HRP, and we can, under bioluminescence microscopy, indeed see that we have light production from within the vesicle. And we can use this generated light to target BCL4, which is labeled with a green fluorophore, to the surfaces of our synthetic cells. But does this work with neutrophils? So neutrophils can be activated through the addition of chemicals. And here we do this with the addition of PMA. And you can see that upon addition of PMA and the luminescent reagents, we can generate lots of photons. And then we can use these photons to recruit BCL4 that we add to solution to these cells to their surfaces. And thereby we can selectively target activated cells using this light-based communication between the synthetic and the living component. So yeah, with this, I hope I was able to give you an overview of some of our work where we take molecular building blocks to build cell-mimetic systems. I have not really touched on too much on our program on the assembly of multicellular systems, where we use mammalian cells, bacteria, but also we produce biohybrids to generate tissues, biofilms, and with very precise structures. But maybe this is a topic for another day. With this, I would like to really thank my team. It's a wonderful mixture of chemists and biologists from all over the world. They like shooting light at all types of cells and at each other, and we are having fun with this. I also would like to thank my many collaborators that allow us to go beyond what we are able to do in the lab alone, and the many funding bodies that support this work. And you for your attention. Thank you very much.
Thank you so much for this really interesting talk and an introduction into the power of such photomimetic systems for modulating and controlling synthetic cells. And it was also really interesting to see at the end how this can also be used to control for the crosstalk between synthetic and actual cells. It would be interesting to know how this will then develop, how the story continues into building into tissue. So as you said, the next talk,
yes,
we hope to hear the next part.
I promise that I give an update on that soon.
How these cells come together to make biofilms, tissues. Yes, as I mentioned at the beginning, for those who have questions, please type in the Q&A. And before that, I just want to, because as a moderator, I'm going to take the privilege of asking the first question at the beginning, where you talked about how these signals can be for switching the cells. I also wanted to know if it is possible to modulate the amplitude of the outcome. If you have more of the probes, will you get a brighter outcome with the light? Can you correlate it? Or
yes,
is it more a yes and no, a binary system?
No, you're absolutely right. Depending on how much substrate we add, we can control how much light signal we get. And depending on how many photons we are generating, the message is stronger or weaker, and that also impacts the action. We have not really quantified this. I just know this from the many optimization experiments that we have done. We are currently working on generating a more quantitative model of this light-based communication, also answering what the signaling range for such a communication would be. And so, yeah, this is work in progress. And yeah, we need more structured methods for this, because currently what we have is all in solution. So we have random distributions of the sender and receivers. And so get a statistical response. But to answer this question, we really need to have precise distances between sender and receivers. And yeah, we are working on that.
Because that could also have a lot of applications and
yeah.
Okay.
And I mean, what we also know from our direct blue light activation studies is that the response very much correlates with light intensity. So you can modulate the response certainly through light intensity. So this part we definitely know already.
Okay. The first question from our audience is along the same lines about the order of introduction of the ionophores, where you said that has an influence. Does the concentration also have an influence?
There we actually see that it's really more binary. That we have optimized the concentrations so that we get maximum response, there it's really, I mean, either it's a very small window, but already doubling the concentration usually gets us to the complete on state.
Okay. What is the composition of the GUVs?
Yeah, it is a very important question. So we try to keep the lipid composition as simple as possible. So most of these are simple DOPC or DOPG vesicles. For our adhesion work, we add about 10% negatively charged lipids so that we have some electrostatic repulsion and don't get a high background in the clustering. For some work, we need to add a little bit of cholesterol, especially when we are interfacing the vesicles with living systems so that they are more resistant, basically, and mechanically more stable. And sorry, and one final lipid that I should absolutely not forget is that we use DGS-NTA for anchoring our photoswitchable proteins to the surfaces of our synthetic cells. And there, of course, we also optimize the concentration of that one.
Okay. The next two questions are along the same line. Can you comment on how heat and temperature would affect these light experiments, especially given that light generates heat?
So we do all our synthetic cell work mostly at room temperature, but we also have used the same photoswitchable proteins in cell culture conditions, so at 37. And not just us, like there's a whole optogenetics community that uses these photoswitches at physiological temperatures. And so temperature per se is not a problem for the protein part. The luminescence reactions actually do not generate substantial amounts of heat. I think at this point, maybe I should once again make clear how little light we need. So these proteins respond to light intensities available on our planet. And so in my group, when people are working, the lights are off and we have dark blinds. And so we have to really take care that we do not photoactivate our system. So very little light is sufficient and we do not generate heat by illuminating. But we always do the controls there, yeah.
The next is a follow-up to the lipid composition. Does this membrane composition mimic neutrophil cell membrane when it comes to application?
For the synthetic cell, no, not really. We have just, so for this BCL4 protein, it's also described in literature. The origin of the charge is not important. It's mostly about charge density. So as long as 20% of the lipid is -1 charged, this BCL4 will bind in a light-dependent fashion. And so if you use a triple negatively charged lipid, like just DGS-NTA without a metal coordinated, you can get away with 7 to 8%.
The next question is, have you observed any tolerance to signal from non-synthetic cells?
I'm not quite sure how I should interpret this. I think as long as the synthetic cells are stable under cell culture media conditions, they could perceive signals from living cells. And I think the example that I showed with the activated neutrophils show that, yeah, the neutrophils can generate light signals and then the synthetic cells can react to them.
Okay. Then I think with that, we'll close the session.
Oh,
And thank you so much, Serafina, for taking the time and sharing your work with us and for the rest of the audience. And for all the attendees, thank you for joining us. And we hope to see you next month with another edition of the Matter to Life lecture. And we hope to have Serafina again, maybe next year, with new results and with an update of this story.
yes. Yes. Thank you very much for having me. It was a pleasure holding this seminar. And yeah,
Thank
see you
you.
next time. Bye.